Hi, today we're going to be looking at enzymes, how they function, what they are, their shape, different things that can affect them, and things called inhibitors. So we're going to start off by looking at this sucrose molecule and how it breaks down. This right here is the sucrose molecule. And as you can see, there's two major parts, this side on the right here and this side on the left. Now it breaks down into two things called glucose and fructose. Sucrose is a form of stored energy that the body uses. So when it doesn't readily need glucose, it can combine it with fructose and create this storage cell, this sucrose. And so when the body needs more glucose, this sucrose has to be broken down. Now this reaction would normally take years for just a small amount of sucrose to be broken down on its own. So it gets some help from this little molecule right here called sucrase, which is an enzyme. It acts as a catalyst, speeding up the reaction. That's important. An enzyme speeds up a reaction so that thousands of sucrose molecules can be broken down in just a matter of seconds. So this is actually what is happening when an enzyme is used. It alters this activation energy right here and changes how much energy it takes a reaction to take place. So right here is the energy where the reactants are. This right here is how much energy it takes to start the reaction. And then for this specific exothermic reaction, energy is released. So this energy right here determines the speed the reaction takes place. It determines how many molecules can react at a certain pace. So what an enzyme does is change that needed energy, like right here. And so this difference right here is how much energy is saved when an enzyme is used. This speeds up the reaction greatly. And so many reactions in our body would take too long to occur on their own. But because of these enzymes, the reactions move forward at the pace our body requires, keeping us healthy. So now we're going to talk more about how enzymes actually work and what they look like. And you can kind of think of it as a lock and a key. So right here, you have the lock. And only one key out of all of these can fit inside that lock. If the other key tried to fit in there, it wouldn't work. It wouldn't unlock the lock. And so enzymes are kind of the same way. An enzyme is like a lock. Only one specific molecule can bind to that lock. And so any other molecules ignore the enzyme, and enzymes ignore other molecules. They're made specifically for one certain type of reaction. Sucrase can only bind with sucrose, and that's its only function. So here's a visualization of that sucrose-sucrase reaction we talked about earlier. Right here is the enzyme, the sucrase, and here is the sucrose, the molecule. And so you can see that they bind perfectly together. And so no other molecule would have that exact shape and would be able to bind to that sucrose. This diagram also helps us look at vocabulary that goes along with the enzymes. Right here is the active site, which is just where a molecule binds to an enzyme. A molecule is called, in these types of reactions, a substrate, which is anything that binds to an enzyme. Once they're bound together, like in this graphic right here, they're called an enzyme-substrate complex. And then, after they're bound, the products are released. Right here, glucose and fructose. And then, and this is important, the enzyme is ready to be used again. Enzymes don't get used up in their reactions. They can be used over and over and over again, which really helps with the speed that we talked about earlier. So some key things to remember is that a substrate binds to an enzyme's active site and creates the enzyme-substrate complex. It's easy to remember because if you remember the enzyme and the enzyme-substrate complex is just them bound together. So here's another important thing to note. An enzyme is a specialized protein. And if you remember about proteins, they're just made up of a bunch of little amino acids. That's all this is. This is what an enzyme actually looks like. It's not as perfectly shaped as this one looks. It's actually just a clump of amino acids and not much else. And so even though this right here is what you'll normally see, keep in mind that this is what it actually is. It's a protein, a specialized protein. And so here are a couple of things that can affect how an enzyme works, how well it works, how much energy it can decrease a reaction. So they only work at their speeds in certain conditions. Things like temperature, pH, and salinity, which is just the amount of salt in the body, can all affect how an enzyme will work. And so here you can see how temperature and pH affects an enzyme. And so right here is the optimal point. That's the point where the enzyme can take as much energy off of a reaction as possible. And so as the temperature decreases and increases, this amount of energy slowly decreases until it stops working. Same thing with the pH. Right here is the optimal pH point, and as it changes, as it deviates from that optimal point, the enzyme isn't as effective. So what you can see is that enzymes require a very specific range of conditions to function. Even just a small decrease in pH or a small decrease in temperature drastically changes how fast a reaction can take place. And so this is why the body, for example, requires a very specific temperature, why our blood requires a very specific pH. It's because these reactions that are required for the body to survive have to work at a certain speed. And that speed requires the enzymes that require a specific condition. So it all works together, and it's all to ensure that your body is healthy. Lastly, we have to talk about inhibitors. So earlier I said that only one specific molecule can bind to an enzyme. Now, that's true for the most part, but there are also these things called inhibitors right here that can fit to the enzyme but don't do anything. And so what these do is, this is what a reaction normally does. The substrate binds to the enzyme and products are released. But these inhibitors can bind to the active site, preventing those substrates from binding, and so that reaction can no longer take place. The enzyme is null and void. The enzyme no longer works. And so it effectively slows down the reaction. And so that's about it about enzymes. Just remember that enzymes speed up a reaction, enzymes are specialized proteins, enzymes require a very specific range of conditions, and enzymes can be stopped by these inhibitors.